In chapter 15, we make a connection between inherited characteristics and those chromosomes that are found in the nuclei of cells. From chapter 14, we remember that Mendel discovered how characters are passed on from generation to generation. And he was able to formulate 3 different laws of inheritance that were called the law of segregation, the law of dominance, and the law of independent assortment. In the law of segregation, he postulated that there were heritable factors in cells, in somatic cells, and in those somatic cells, those factors were present in 2 copies. And he also realized that gametes, eggs and sperm, only had 1 copy of those factors. And the part of the segregation comes from that he understood that in order to make the gametes, you would have to separate the 2 copies of the factors in the process of making the gametes, which later became to be called meiosis. Now, the law of dominance is about the interaction between the factors in an individual cell or in an individual organism. If you have an individual that has 2 different copies of Mendel's factors, and what that means in modern language, if you have an individual that has 2 different alleles of a gene, then only one of those is going to be manifested, is going to be shown in that individual's physical appearance. And that he saw with all of his pea plants. If the pea plant had 1 factor determining purple flower color and 1 factor determining white flower color, only the purple color would be seen in the plant. So that one was the dominant factor. If it had 1 factor, 1 allele determining tallness and 1 allele determining shortness, the plant would be tall because tallness was dominant over shortness. So that his law of dominance decreed that one of the 2 alleles is going to be dominant over the other. And how do you call the one that is not seen in that individual that carries 2 different copies? That would be the recessive allele. Another thing that he realized is that factors that are governing different traits are independent of each other. In other words, the color of the flowers of a pea plant is completely independent of the color of the peas of a pea plant or of the height of that pea plant. These things they can be inherited by an individual plant in any combination. In other words, you can have a plant with purple flowers that is tall and has peas that are green, or it can have white flowers and peas that are green and be tall, or any other combination. That became the law of independent assortment. So he realized that those factors are not linked together, they assort independently. And we're going to look in this chapter at the explanation, the physical explanation for all of these observations and laws that Mendel formulated. Another thing that he understood and that he wrote down in his observations is that the factors are discrete and particulate, meaning they're not liquids that blend and mix together. At the time that he was doing his experiments, the concept of blending inheritance was very popular. And, it was thought, well, if you have a plant that has purple flowers and one with white flowers, and you mix them, then maybe you get like a light purple or a pink color. But he demonstrated that is not the case, and he also demonstrated that even though the white color is not visible in an individual that carries both characteristics, it hasn't been affected in any way, and it can come back in a later generation. So in the coming slides in this chapter, we're going to see what what is behind these observations that Mendel left with us. Unfortunately, people didn't really pay attention to Mendel when he published his observations. He died basically unacknowledged, and his work wasn't known until it was rediscovered in 1900 by 3 scientists at the same time. In 1902, Sutton and Boveri proposed the chromosome theory of inheritance. In that theory, they connected Mendel's observations of his factors with the visible behavior of chromosomes visible under a microscope. And when we look at it with 20/20 hindsight, we realize that genes, Mendel's factors, are physically located on the chromosomes. And observations that Mendel recorded, such as segregation, the separation of the factors during the production of egg and sperm, that is the separation of the pairs of the members of each pair of homologous chromosomes during meiosis I. In this picture, we're going to have the opportunity to see that connection between Mendel's observations and the behavior of chromosomes in cells. So we're going to look at both things at the same time. On the left, on top here, we have the P generation, the parental generation, and that parental generation is going to be represented by these two pea plants, one of them which has yellow and round peas over here, and the other one has green and wrinkled peas over here. So we're looking at two characteristics at the same time. The genotypes of these pea plants, the one with yellow and round seeds, is big Y, big Y, big R, and the genotype of the other plant is little y, little r, little r. We know that big Y is dominant over little y. Big Y stands for yellow seeds, and little y stands for green seeds, and big R is dominant over little r. Big R stands for round or smooth seeds, and little r stands for wrinkled seeds. Both of the plants in that P generation are true breeding, and true breeding means that they are homozygous for those two characteristics. So they're homozygous for the Y character, for the color of the P. In the plant on the left side, it is homozygous for the yellow color allele, and it is also homozygous for the round shape allele. The plant on the right side is homozygous for the green color allele, and it is also homozygous for the wrinkled shape allele. Now, if we take a look at a cell in the body of that plant, we're just looking at the chromosomes that carry those two characteristics, the shape of the P and the color of the P. And those two characteristics are carried on two separate pairs of chromosomes. So we have a long pair of chromosomes that carries the shape characteristic, the R's, and a short pair that carries the color of the P characteristics, the Y. And one of the cells, the one on the left, has red chromosomes, and the one on the right has blue chromosomes, so we can keep track of them. And let's just make sure that we see what's going on. So the big pair of chromosomes, the big pair of red chromosomes, is carrying capital R on each of them. So capital R here, capital R there. And the small pair of chromosomes is carrying capital Y. On the other side, the big pair is carrying little r, lowercase r, and the small pair is carrying lowercase y. So we cross these two plants together, but before we can do that, they have to go through meiosis. So meiosis happens first, and in meiosis, as you know, we're going to cut in half the number of chromosomes. So the plants produce gametes, each of which has only one copy of each chromosome. So the cell on the left has one copy of the large chromosome and one copy of the short chromosome, one copy of capital R and one copy of capital Y. So those are its gametes, capital R and capital Y. The plant on the right has the gametes little y and little r. Okay, now we're going to take a look at the first filial or F1 generation. In that generation, we're going to form zygotes by combining the two gametes from the P generation. So we're combining this cell and that cell. Let's say that the one with the blue chromosomes produces sperm. So if this is a sperm cell, the other one is an egg cell. Then now we combine the sperm cell with an egg cell and we generate zygotes. And all of the zygotes are going to have the same genotype, the combination of the information in the sperm and the egg. So they have big Y, big R, little r. That's their genotype. The plants of this generation are heterozygous or hybrids for both characteristics. They've got two copies of each chromosome, just like their parents did, the somatic cells of their parents or the germline cells of their parents, two copies of each chromosome, but in their case, they're two copies that carry different alleles. So they've got a big R and a little r, and they have a big Y and a little y. Now meiosis is going to happen on these plants. And in the next comment, we're going to take a look at what happens there. The cells that we saw here are equivalent. It is really the result of the fertilization from these two cells. Two of these cells produce one of this type of cell. And what they're showing us, you could have from this fertilization, from the fertilization between sperm and eggs in the P generation, you can make a lot of individuals in the F1 generation. Just like if you cross one pea plant with another pea plant, you're going to get a lot of peas produced and a lot of baby plants. So let's just say this was one of those baby plants and this was another one. They have the same genotype. Both of them are big Y, big R, little r, and the peas are both yellow. So what we're going to take a look at is that they're going to undergo meiosis, but we're looking at two different possibilities of meiosis in these plants that have the same genotype. On the left side, we see that the chromosomes have by chance arranged themselves in such a way that both of the blue chromosomes are on the right side of the cell and both of the red chromosomes are on the left side. when we look at the same situation of metaphase 1 on the other cell, we see here it's different. Here you've got on the right side of the cell, you've got big R and little y, and on the left side you have little r and big Y. Those two combinations are equally plausible, equally possible, and if you have lots of cells that are undergoing meiosis, you're going to have all of these combinations occurring. And that is the law of independent assortment, that the combination of R and Y can be any combination. So in the end, after meiosis is finished, these are going to be your final products. So one of the cells is going to have big R and big Y. So these are gametes. So it's going to have the two dominant characteristics. Another one is going to have the two recessive characteristics, little r and little y. Another gamete is going to have little r and big Y. And a fourth gamete is going to have big R and little y. Those are all the combinations of R and Y that you can have. That is independent assortment. It doesn't matter if you have a capital R and a capital Y, or a capital I, R and a little y together. As long as your gamete has one R and one Y, you can have any possible combination. Notice that none of these gametes has a combination that's like RR or YY. It is exactly one R and one Y, and you need to have one of each. So let's take a look also at the law of segregation. Where do we see that? Well, we see that in the first meiotic division, when the members of the homologous chromosomes separate, we have two pairs of homologous chromosomes. So this is one pair, the pair that carries the R's, and the pair that carries the Y's is another pair. They separate from each other in meiosis 1. Same thing happens in the other cell. So that is the law of segregation, where you go from having two copies of a given allele in one cell to having just one copy of a given allele, one copy of each chromosome per cell. When the chromosome theory of inheritance was proposed by Sutton and Boveri, a lot of people were skeptical about it. And one of those skeptical people was Thomas Hunt Morgan. He was a geneticist, and he worked at Columbia University, and he started working with a little fly called Drosophila melanogaster. And in the next few slides, we're going to take a look at how he accumulated evidence that supported the chromosome theory of inheritance. And this was actually to his own surprise, because he didn't believe in it and he was going to debunk it. That is one of the, I'd say, that is the beauty of the scientific method. If you are doing your experiments correctly, and if you have an open mind, sometimes you have to be ready to abandon your previous conceptions and change your mind about, change your opinion about the thing that you are experimenting with. What were the advantages and disadvantages of using the fruit fly Drosophila melanogaster as a model to do studies in genetics? Well, one of the main advantages is that they were free. You can find fruit flies around rotting fruit and around the trash, and if you set out some traps for them, like a piece of banana, and with a net, you can catch them. And another thing is that they develop fast. You can go from egg to adult fly that lays new eggs in from 9 days to 2 weeks. So that's a very fast generation time. They're also easy and inexpensive to keep. You can use an old milk bottle. At the time that Morgan was doing his experiments, that's what they would have used, those old glass milk bottles, and you fill them with a growth medium that has bananas in them and agar to make it a solid medium, and you put some yeast in it and some paper for the flies to walk on, and there you have it. You have a culturing medium for your flies. Some disadvantages. Drosophila had not yet been developed as a model. So that means there were no strains of Drosophila that he could order and that were guaranteed to be true breeding. They also didn't know anything about those flies, so they didn't have any references. They were starting in uncharted territory. So more information about Drosophila melanogaster. As you can see, it's a very handsome little fly. They're very tiny, much smaller than a housefly. They have 4 pairs of chromosomes, and of those, 3 pairs are autosomes. Autosomes are the chromosomes that do not carry the sexual characteristics, and 1 pair of sex chromosomes. So the sex chromosomes would be XX in the female and XY in the male. In describing Drosophila, we call the most commonly seen version of a trait, we call that the wild type. So having brick red eyes is wild type in Drosophila. Having long wings that extend past the length of the body is wild type. So a mutant phenotype has a different trait for a given characteristic. So a mutant trait for eye color includes white eyes or sepia eyes or apricot colored eyes or yellow eyes. In our picture over here, we've got flies with all different color eyes. It looks to me like the regular color may be this one, although that's very bright. And here is a white colored eye, and this one is maybe the brown. So you've got different color eyes, and then the wings, all of them seem to have long wings. So this is the normal length of the wings. In this particular one, the wings are extended. That often happens when the flies have been anesthetized too much and they've actually died from the treatment. So they would be in that shape. Sometimes flies have a mutation that makes their wings very small. They would be like this size and crumpled, and those are called vestigial wings. So that's a wing mutation. Other times they have curly wings, so the wings go up like that. And I think they can't fly with those, with the curly wings. Definitely not with the vestigial. After a couple of years of no luck in developing mutations, one of Morgan's students discovered a white-eyed fly in one of the tubes. So that fly was picked out and was carefully mated to other flies, and they checked at what happened with the transmission of that physical characteristic. So he mated that white-eyed fly, which was male, with a female fly that had red eyes. Remember, the red eye color is the wild type. And they observed that the F1 generation, in other words, the offspring from that cross, all the flies, males and females, had red eyes. And then when he mated those flies among each other, he noticed in the second filial F2 generation, he observed a 3-to-1 phenotypic ratio with 3 flies with red eyes for every 1 fly with white eyes. And you remember probably that that is the same phenotypic ratio that Mendel observed for his monohybrid crosses. However, there was one thing, the only flies that had white eyes were male, so it was only present in the males, and the proportions were that half of the males in that F2 generation had white eyes, the other half had red eyes, and all of the females had red eyes. So that's different from saying a quarter had white eyes and 3/4 had red eyes, and you would expect to have a quarter of in that quarter there would be half males and half females, but it wasn't like that. So Morgan concluded that eye color was a sex-linked trait and that it was recessive. And in the slides that are coming up, we're going to see in illustrations how that works. After a couple of years of no luck, let's take a look at how sex and eye color, sex meaning gender, and eye color are related in Drosophila. Drosophila female flies have 2 X chromosomes, while males have 1 X chromosome and 1 Y chromosome. Remember, the X and the Y chromosomes are the sex chromosomes. And it turns out they're not only the sex chromosome, they also have other genes, just like our X chromosome, our human X chromosome has other genes that are not only related to sexual characteristics. So eye color is on the X chromosome, and the white mutation, the mutation that gives the white eye color, is a recessive mutation. In general, a recessive mutation is one that needs to be present in both copies of a chromosome in order to be manifested in the phenotype of the organism. In other words, in this case, in order to be seen as a change in the eye color of the flies. So if you have a female that is heterozygous, meaning that she has 1 regular normal X chromosome, which is denoted as X plus, and the other one is denoted as XW. So it has, that X chromosome has the white mutation. That heterozygous female will have red eyes because the white mutation is recessive, and her normal chromosome, the allele on her normal X chromosome, is dominant over the recessive mutation. A male, however, only has 1 copy of the X chromosome. So if he happens to get the XW X chromosome, the X chromosome that carries the mutation that makes the eyes white, he's going to have white eyes. He doesn't have any other chromosome or any other allele that compensates. So he will have the white eyes, and he will express that recessive mutation. So now we're going to take a look at the experiment that Morgan did, and this is the first part. So this was the special fly, the male fly with the white eyes. This is the symbol for male, and this is a symbol for female. So a male fly with white eyes was mated to a female with red eyes, and in the F1 generation, first filial generation, all of the offspring had red eyes, whether they were male or female. If you then allowed those flies from the first generation to mate amongst each other, then you get this arrangement of eye colors. All the females have red eyes, and among the males, half of them have red eyes and half of them have white eyes. This is in the F2 second filial generation. Now let's combine the information that is from the physical characteristics, from eye color, with information from chromosome behavior. So let's take a look. We have the P generation here on top, the parental generation, and the female, her chromosomes look like this. She has 2 X chromosomes in red, and both of them are W+. So she has 2 wild-type X chromosomes. Her spouse, the male, has got 1 X chromosome that is white, and 1 Y chromosome. He's got an X and a Y. And the X that he has is with the mutation. This is plain W. So look at his eye color. He's got white eyes. Again, contrast with the female. Both of her X chromosomes are W+. So in the cross, they're both going to make gametes. The female, with respect to the X chromosome, makes only one kind of gamete. So in this crossover here, when she makes her gametes, she makes just 1 X chromosome per gamete, and that X chromosome is wild type for the eye color, so it's W+. The male is going to make 2 kinds of gametes, 2 kinds of sperm. Half of his sperm are going to have his X chromosome, and that X chromosome carries the white allele, and the other half are going to have the Y chromosome. So they don't carry any information about eye color at all. So when you make that cross, you're going to have that half of the offspring are going to be female, and they inherit the wild type chromosome from their mother, and they inherited a white chromosome from their father. So 1 W+ and 1 W. And so the result is going to be a female fly with red eyes, because the white mutation is recessive, and the wild type version on the red chromosome makes up for the defect in the white chromosome. Let's look at the males. He gets from his mom, he gets the wild type X chromosome, this one. And from his dad, he gets a Y chromosome. He's going to have red eyes too, because the only X chromosome that he has is the one from his mom, which gives him the wild type allele for eye color. So in the next comment, we're going to look at what happens after that. When the members of the P generation made their gametes, in other words, these eggs and these sperm, the female only made 1 kind of gamete because she is homozygous in the X chromosome. The male made 2 kinds of gametes because he has 1 X and 1 Y chromosome. Now in the next generation, we're going to look at the cross between members of the F1 generation. So these 2 are going to be crossed with each other. So we're going to look at the gametes that are made by this female and by this male. And when you make gametes, what you do is that you're going to separate the 2 alleles of a gene. In other words, you separate the 2 copies of a homologous chromosome pair. So now we're going to separate these two chromosomes from each other. So this female makes two kinds of eggs. Half of her eggs are going to have the red wild type X chromosome, the W plus. The other half are going to have the blue white, the mutated chromosome, X chromosome. The male is also going to make two different gametes. Half of his gametes are going to have the wild type X chromosome that he inherited from his mommy, and the other half are going to have the Y chromosome that he inherited from his dad. Now they're going to have offspring, and we're going to combine these gametes in every possible way. So the first is W plus with W plus, which gives us this one. This is a girl with red eyes, and she's got two wild type X chromosomes. Then over here we have another girl, two X chromosomes, one that has the white mutation and the other one that is wild type, but she's got red eyes too, because remember that the white mutation is recessive to the wild type. Then there are two boys on the right side. The one on the top here gets the Y chromosome from his father here, this Y chromosome from his dad, and he gets this X chromosome from his mom. And his eyes are red because the only X chromosome he has is the wild type with the normal eye color, so he has red eyes. And then finally there's the white boy over here, and he has a white X chromosome, this one, that he got from his mother, plus a Y chromosome that he got from his father. So since the only X chromosome that he has the white mutation, this male fly is going to have white eyes. So that is going to give you what kind of ratio it's going to be. As far as eye color is concerned, it's a 3-to-1 ratio, right? 3 red eyes to 1 white-eyed fly. Let's see how sex is determined in human beings, and that applies to all other mammals as well, except maybe the platypus. So humans, in their somatic cells, have 23 pairs of chromosomes. That makes then 46 chromosomes total. Of those 23 pairs, 22 pairs are autosomes, and 1 pair is a sex chromosome pair. The autosomes are those chromosomes that are the same among males and females. And the sex chromosomes are the ones that are different. In females, the sex chromosome pair has 2 X chromosomes, and in males, it has 1 X and 1 Y. So in human beings, the sex of a child is determined by whether it has a Y chromosome or not. If it has a Y chromosome, it's a boy. If it doesn't, it's a girl. Even if, for example, in an unusual case, if you have a child that has this sex chromosome combination, and this can occur in a condition known as non-disjunction, then it would still be a boy, because the presence of that Y chromosome overrides the presence of the 2 X chromosomes. And that means also that the sex of a child is determined by the male If the male gives an X, it's going to be a girl. If the male gives a Y, it's going to be a boy. Henry VIII should have taken Bio 210. And we see this same sex determination system in other mammals as well. If you take a look here at this picture of the sex chromosome pair in humans, you can see that the X chromosome is much bigger than the Y chromosome, and that you cannot actually see that, but the X chromosome has a lot more genes than the Y chromosome. There are other genes on the X chromosome that are not related to sex determination and that are necessary for survival. So every male needs to have that one X chromosome. On the Y chromosome, there are really no genes that are necessary for the survival of the organism. Otherwise, females would not be able to survive because they don't have the Y chromosome. But there are genes that are important for male fertility on the Y chromosome. On the Y chromosome, there is a small area, a small region that determines the sex, determines the male sex, SRY. It stands for Sex Determining Region Y. This slide shows us in an illustration the mammalian sex determination system, which is also called the XY system. So if we look here at the parents of an individual, you've got the father on the left, and he has 44 chromosomes plus the XY pair. Together that makes 46 chromosomes. And the mother also has 44 chromosomes, but she has an XX pair. And that also makes 46 chromosomes. So these are like germline cells that are going to undergo meiosis to make gametes. In the case of the father, he's going to make two different kinds of sperm cells as a product of his meiotic divisions. And half of them are going to have 22 chromosomes plus one X chromosome, and the other half are going to have 22 chromosomes plus one Y chromosome. In other words, you have a sperm cell with an X chromosome and a sperm cell with a Y chromosome. And then the female, the mother in this cross, when she makes eggs, all her eggs, 100% of her eggs, are going to have 22 chromosomes, the autosomes, plus one X chromosome. In all of these cases, the 22 chromosomes refer to the one set of autosomes. Now, we're going to do the cross, and depending on what the male gives, the sex of the zygote will be determined. In this case over here, the zygote is a female, so that means she got an X from her father, because she always gets an X from her mother. The mother doesn't have anything but X to give in the sex chromosome department. So she gets an X from her mother, and she also gets an X from her father. So this zygote is XX in the sex chromosome pair. And then she also gets 22 autosomes from her father, and she gets 22 autosomes from her mother. So that gives her 44 autosomes plus XX, and that is a human female. Over here is a human male, and he got 22 autosomes plus the Y chromosome from his father, and 22 autosomes plus the X chromosome from his mother. So that makes him 44 autosomes plus the XY pair, and that is a human male. Human beings have 23 pairs of chromosomes. 22 pairs of autosomes and 1 pair of sex chromosomes. In human females, the 23rd pair, the sex chromosome pair, consists of 2 copies of the X chromosome, and those 2 copies of X are homologous. They form a homologous pair, and so they can synapse, they can come together, lie next to each other, and they can cross over during meiosis because they're basically 2 copies of the same chromosome. In human males, on the other hand, the 23rd pair consists of 1 X and 1 Y chromosome. And as you saw on the picture in the previous slide, or the 2 slides ago, the X and the Y are very different from each other in size and in content. However, to make the pairing possible, they do have short areas of homology so that they can recognize each other and pair. And so the X and the Y chromosome do pair together during meiosis in males. There are certain conditions that are said to be linked to the gender of an individual, and they're called sex-linked conditions. And the majority of them are on the X chromosome, simply because the X chromosome has a lot of genes. And the inheritance of these conditions will depend on the genotype of the parents. Let's take an example. A sex-linked gene, an example of a sex-linked gene is red-green color blindness. If you have a cross between a male, a human male that is colorblind, and a female that has normal vision, then all the daughters of that mating are going to be carriers themselves, but with normal vision. That means that they can pass on the condition to their children, but they will not be affected by color blindness themselves, because they will have 1 X chromosome with the mutation, but they have another X chromosome that doesn't have the mutation. And since the mutation is recessive, the quote-unquote wild-type X chromosome will be providing the gene product that is necessary for normal vision. All sons of the same mating, on the other hand, are going to have normal vision. It's kind of interesting, though. Their father is colorblind, but he will have normal vision sons. And that is because the sons did not get his X chromosome, they got his Y chromosome, and there is no equivalent to that gene on the Y chromosome. And the sons got the X chromosome with the normal allele from their mother. So a colorblind male and a female with normal vision who is not a carrier herself, they're going to have daughters that are carriers and sons that are completely normal. Let's look at 2 other possible combinations. Here we have a male who has normal vision and a female who is a carrier. So she is heterozygous for the color blindness allele, has 1 normal X chromosome and 1 with the color blindness allele. The male has normal vision, so his 1 X chromosome has the normal allele. In that mating, each of their daughters has a 50% chance of being a carrier, meaning that she won't have the condition herself, but she can pass it on to her children. And each son has a 50% chance of having color blindness. If, on the other hand, we have the 3rd condition here, that the male is colorblind, so he's got the defective X chromosome, and the female is a carrier for the condition herself, but she has normal vision, so she's got one mutated X chromosome and one wild-type X chromosome. In this case, the daughters will have a 50% chance of being carriers for color blindness and a 50% of being colorblind themselves if they inherit the X chromosome with the allele that determines color blindness from each of their parents. And then the sons will have 50% chance of being colorblind and 50% chance of having normal vision. This slide shows us the three conditions that we looked at in earlier slides. The first one was the mating between a colorblind male and a woman with normal vision who is not a carrier for colorblindness either. Let's look at the guy first. He has got an X chromosome and a Y chromosome, and that X chromosome has the mutation, so it's got the color blindness mutation on it, and he is colorblind. And they're also showing us the colorblind individuals have the sort of a dark orange, and then the carriers are like an intermediate pink, and the ones that are not affected are not colored in at all. So he is marrying this woman who has normal vision, X capital N, X capital N. And now we take a look at their children. Now first take a look at their gametes. The male is going to make X chromosomes that have the mutation. That's all he has. And he's going to have Y chromosome in his gametes. The woman is going to have eggs that all have the normal X chromosome. So when they mate, you've got zygotes that are going to be, half of them are going to be girls, and the girls are both going to be carriers for the condition. They have each one normal X chromosome and one mutated X chromosome, so they don't have the condition themselves, but they are carriers and can pass it on to their children. The males are normal with respect to color vision. They have normal color vision. They have only one X chromosome, which they got from their mother, and she gave them a normal version of that allele, and then from their father they got the Y chromosome. So they don't have the disease themselves, and they can also not pass it on to their children. In the second situation, we've got a woman who is a carrier for the disease, mated with a man that has normal vision. So she is a carrier, which means she's got one normal allele and one mutated allele. He has only the normal allele in his one X chromosome. So again, we make the gametes for each of them. He makes sperm that have an X chromosome that has the normal allele, and then he has sperm that carry the Y chromosome. She has two different kinds of eggs. Half of her eggs carry the normal allele of the condition, X capital N, and the other half carry the mutated version, X lowercase n. So now let's take a look at what kind of offspring we get. Again, of the four, two are girls and two are boys. Among the two girls, this one in the upper cell of the table is completely normal with respect to color blindness. She inherited the normal allele from her father and also from her mother. The second girl inherited the normal allele from her father over here, and she inherited the mutated allele from her mother. So like her mother, this girl is a carrier for color blindness. She is not colorblind herself, but she can pass the condition to her children. And now let's look at the boys in that mating. The boy on the top has normal color vision. He's got the normal X chromosome and a Y chromosome as his sex chromosome pair. And the boy in the lower right corner has color blindness. He's got the mutated X chromosome that he inherited from his mother and the Y chromosome that he inherited from his father. So you see, in this particular case, we have a child that's a carrier and a child that has the condition, and the two parents, one of them is a carrier and the other one is completely free of the condition. So phenotypically, both of these parents have color vision, but they have a child that is affected, or they could have a child that is affected by color blindness. Finally, in the third condition, the woman is a carrier. She's got one affected allele and one unaffected, a normal allele, and the man has color blindness. His one X chromosome is the mutated one for color blindness. Sperm and eggs made. His sperm cells, the ones that carry an X chromosome, are also going to carry the mutation for color blindness, and then he's got the Y chromosome that doesn't have anything to do with that at all. Then come the eggs of the woman. She makes half of her eggs are going to have the normal X chromosome that will give normal color vision to her children, and the other half are going to have the colorblind allele on the X chromosome. Let's take a look at the children. First, the girls on the left side. Two girls. The one on top is a carrier and the one in the bottom is colorblind. So this colorblind girl, because the condition is recessive, she must have inherited it from both parents. She must have inherited the allele for colorblindness from both parents. And then finally, we've got the males. 50% of them are completely normal with respect to color blindness. So that's X capital N to the capital N Y. And the other 50% has color blindness because their one X chromosome is afflicted with the allele that codes for color blindness. Two more examples of X-linked, sex-linked conditions in humans. So notice that when they think of sex-linked, it usually means X-linked because the X has most of the genes. So they are hemophilia and Duchenne muscular dystrophy. Hemophilia is caused by the absence of clotting factor 8, and the gene is located on the X chromosome. If that clotting factor isn't working, you get very heavy bleeding from minor cuts and bleeding that doesn't really stop very easily, and not just external bleeding but also internal bleeding. There are actually two major types of hemophilia. Hemophilia A, which is caused by the lack of factor 8, and hemophilia B is caused by the lack of factor 9, and that condition is also X-linked. The other condition is Duchenne muscular dystrophy, and in Duchenne muscular dystrophy the protein dystrophin is not made correctly, so it is not available, and it causes a weakening and atrophy of the voluntary muscles that is progressive, and it usually causes the the confinement to a wheelchair of the individual, maybe by the age of 12 or so. It's a very serious condition. And so in both of these cases, you will find that the great majority of the victims are the sufferers of these diseases are going to be males, because with their having just one X chromosome, they will express whatever genes are on that X, whether the condition is dominant or recessive. In these two cases, they're both recessive conditions. So if a female has one mutated X chromosome, her normal X chromosome can make up for the deficiency. In earlier discussions, we've seen the terms homozygous and heterozygous, and we said that an organism or a cell that has two different alleles for a given character is heterozygous. So, for example, a woman who has one color blindness allele on one of her X chromosomes and one for normal color vision will have normal color vision because the condition is recessive and she is heterozygous for that condition. She's also a carrier for color blindness. She doesn't have it herself, but she can pass it on to her children. Notice that this cannot happen in males. A male cannot be asymptomatic. He cannot be a carrier and not know that he is a carrier of any sex-linked condition. So a human male either has the condition, he either has the disease or the condition in his phenotype, so he is colorblind, or he has hemophilia, or he has Duchenne muscular dystrophy, or not affected at all. So if he is not affected at all, it means that he doesn't have the disease and there is no danger of him passing on the disease to his children because it is not in his genome at all. And this happens because males are what we call hemizygous for any condition that is linked to the X chromosome. Hemizygous means that you have just one copy of that chromosome, so any gene on that chromosome is going to get expressed, whether it is a recessive gene or a dominant, whether it's a recessive allele or a dominant allele. So males only have one X chromosome and they're going to express all the genes on it, regardless of whether the genes are dominant or recessive. Women, on the other hand, have two X chromosomes, so it is possible for them to be carriers for recessive conditions, and all carriers are heterozygous for the condition, and they can be completely asymptomatic. They may not even know that they are carriers. An interesting fact about having two X chromosomes is that in mammalian females, in every cell, one of those two X chromosomes is going to be randomly inactivated very early in the development of the female embryo. And then once that X chromosome is inactivated, all the daughter cells that result from that cell through the development of the baby are going to maintain that same pattern. In other words, it's going to give rise for an organism that is a mosaic for the X chromosome. In some cells, the X chromosome that came from the mother is inactivated. In other cells, the X chromosome that came from the father is inactivated. And that gives you a certain patchiness. Now, usually you can't see that in the phenotype, but there are exceptions, and one of those is shown here. This is a tortoiseshell cat, and she is a mosaic, and it's visible that she is a mosaic because the some of the genes for fur color are actually present on the X chromosome. So she's got two X chromosomes. One of them has an allele that codes for an orangey fur color, a sort of a ginger orange color, and the other one codes for black fur. In the early embryo then, at some point in all the cells, one of the X chromosomes gets inactivated randomly. So here you have an example of the cell on the left. You have that the version that got inactivated is the one that is responsible for the orange fur color, and it gets condensed. You see, it's like in a little ball. This is called a bar body. And then in the other cell, it's the other situation. The one that got inactivated is the one that is responsible for the black fur. So in either case, the one that is inactivated is not going to have their genes expressed. So if you inactivated the orange fur in the left, then that's going to give rise to black fur on the cat. If you inactivated the black one, as you have on the right side, it's going to give rise to a patch with orange. So you have all these wonderful gradations and patches in this cat, some having orange fur and some having black fur, and each individual cat is going to be unique for that particular pattern, just as each woman is going to be unique in which of her X chromosomes from mom or from dad is going to be inactivated in each of her cells. So why do we need to inactivate one of our X chromosomes? Well, we saw earlier that an X chromosome actually has a lot of genes that are essential for survival and that are not at all related to sex determination and sex differentiation. Males only have one copy of the X chromosome. That means that that one copy of the X chromosome must produce enough quantities of all the gene products that are needed from that X. Well, sure, that happens. So males are fine with their one copy of the X chromosome. But females have two copies of the X chromosome. So that means that females actually have an overdosage of the X chromosome because they have two copies of it. And in genetics, having too much can sometimes be just as bad as not having enough. So you can't go around with having twice as much of all the genes that are present on the X chromosome. So what females do is to inactivate one of those X chromosomes very early in development. So when a female organism is going to reproduce, is going to have children, that inactivation is going to have to be reversed again, so that the early embryo can express all the genes on the X chromosome, and then a new pattern is going to be established early in the development of the child. So you have something called de-differentiation and reprogramming, in which you're going to turn off all the marks that say turn off this chromosome, and then a new wave of inactivation is going to happen in the new organism. So we saw earlier that in mammalian females, one of the X chromosomes is going to be randomly inactivated in each somatic cell. And when that happens, it's actually going to get twisted and condensed as a very small pellet, and that is known as a bar body. The great majority of the genes on the bar body are going to be inactivated, and this occurs through a process known as DNA methylation, or the addition of a methyl functional group to the genes, to the DNA on the X chromosome that's going to be inactivated. There is also going to be a coating of the whole chromosome with an RNA molecule. This is a very unusual thing. So there is a gene on each X chromosome that's called the XIST gene. And that XIST gene doesn't code for any proteins, but it codes for an XIST RNA molecule, so an RNA transcript. When the X chromosome is chosen to be inactivated, the XIST gene is activated on that chromosome and produces that transcript, many copies of that transcript that attach to the X chromosome and completely coat it. And they attract these other proteins that then methylate the DNA and cause further condensation of the chromatin of the X chromosome and completely twist and roll it up into a little ball that is known as the bar body. So on that X chromosome, basically the only gene that is active is the gene that codes for the XIST, for that RNA transcript. Notice also that here we have an example of a gene that is an active gene, but it does not code for a protein. It codes for that RNA molecule, and the RNA molecule functions as an RNA molecule. It doesn't have to be a protein. This is a picture of a blood smear from a female dog, and like humans, dogs have in their blood, they have a lot of red blood cells, and those are all these cells here. Red blood cells do not have a nucleus at all, so they don't have any DNA. They lose the nucleus during the process of their development. The cell that we are interested in is this cell, a white blood cell. It's called a neutrophil, and the part that the arrow is pointing at is the bar body. You see this little tag here is the bar body, and it is also called, colloquially, it is called a drumstick because of the shape. So when the cell is in interface, that bar body is going to be protruding from the chromatin, and you can see it as a separate little tag. In some cases, that characteristic is used to check for gender. Like in the Olympic Games at some point, this was used to check if people that were competing as females, if they were indeed females.